I'm Paulette Bloomer. I'm a professor of genetics in the Department of Biochemistry, Genetics and Microbiology, and I'm also a member of the Mammal Research Institute in the Department of Zoology and Entomology at the University of Pretoria. Since I started my research group, my passion has been to work on threatened or endangered species or species that are utilized in South Africa for a variety of reasons. But the main thread that I've gone through the work has been conservation genetics. We work very closely with national parks and the provincial conservation authorities because it is important for us that the work is interesting in its own right, but we also want to make a contribution in South Africa, and we want to make sure that the data that we generate can be translated into easy to understand and also practical management actions that the conservation authorities can take to make sure that we protect the rich biodiversity of our country. What our research is showing using genetic tools is that often this diversity is an underestimate of what we actually have on the continent. And South Africa is one of the centers of this diversity, and some of the species are endemic to South Africa, for example, the Cape Grysbok. And why we wanted to do this research on the Cape Grysbok is because of its interesting distribution. It is largely confined to the Cape Fold Mountains, which is where we have the Cape Floristic region. And so by understanding the Cape Grysbok and its narrow distribution that is largely confined to the fynbos, we can understand that habitat association and then also understand what can we do in future to protect the species as that habitat will change based on climate change predictions.
My name is Deon de Jager. Right now I'm a postdoctoral fellow at the University of Copenhagen in Denmark, where I'm still studying African antelope genetics, but this time I'm using ancient DNA, which is DNA from fossils, to compare the ancient populations of South African antelopes to the modern populations of these antelopes. And one of the species that I'm working on is Cape Grysbok, which is why I'm so excited to be involved in this initiative with 1KSA and DIPLOMICS in generating a high-quality genome for the species. Firstly, because there's very little genetic data that has been generated for the Cape Grysbok. And secondly, because it has an interesting biology. The core of its distribution is within the Cape Floristic region. We are responsible for conserving this species, and we have to basically try and figure out why is it adapted to this region while its sister species are not. So there's another species of Grysbok called the Sharp's Grysbok, which occurs in the northern parts of South Africa and further north into Africa. And in between those two distributions, in South Africa, there's no Grysbok species occurring. So we understand why is that, and then what makes the Cape Grysbok or maybe the Sharp's Grysbok unique in their adaptations to different regions or different habitats rather. But now generating data for the nuclear genome, we can really figure out what is so unique about this species in terms of its adaptation and its distribution to the Cape Floristic region.
I'm Anri Van Wyk. I'm a postdoc in BGM, which is a department in the Faculty of Natural and Agricultural Sciences here at the University of Pretoria, and I'm in a group called MEEP, which is short for the Molecular Ecology and Evolution Program. So my current research that I'm busy with is mostly focused on conservation genomics and genetics. I focus on large vertebrae, mostly bovids. The main thing, most of the animals or species that we work on, it's quite novel, nothing has ever been done on them, so it's so important that some of these small antelopes like the Cape Grysbok, to sequence that individual and have long read sequencing for us to do. We can determine genetic diversity, we can look at runs of homozygosity if the individuals are inbred, and all of that can then be brought back to a proper conservation management plan for the species.
Over this period, and now with the advantage that we have through DIPLOMICS and the 1KSA project, is that we can, for the first time really, as South African scientists, contribute to the global genome sequencing effort. And I really think that this project is very important to show the contribution that we've made over a long period of time in terms of conservation genetics, but now we can really move it to conservation genomics. And it's not only just about us making our mark on our unique biodiversity, but also about the value of conservation genomics. We can look at the link between different populations. As Anri mentioned, we can study runs of homozygosity and estimate individual levels of inbreeding. The concept of inbreeding is a well understood and accepted concept in conservation genetics, but the concept of outbreeding has been very controversial. Through genomics, we can really tackle this issue because for the first time, these tools allow us to really understand adaptation and then to look at the potential breakdown of adaptation when we have hybridization and integration between closely related species. So this is a really big leap forward for conservation in South Africa, but that does not take away from our ultimate goal, and that is to translate these findings into conservation actions.